Hello friends, welcome back to my channel Medicine 101. Today I will talk about glycolysis part 2. Okay, before starting the video, let me tell you guys, I have already uploaded the video in terms of glycolysis part 1, where I have discussed how the glucose that will be taken within the cells. There are two mechanisms. One is facilitated transport, another is active transport. So before watching this video, I request you guys, please watch the first video, that is in terms of glycolysis part 1. If you watch the first video, then it will be easier to understand. Okay, so let's talk about these things. So in this video, basically I will talk about whenever the glucose that will be taken within the cells, then the glycolysis process that will be started. Whatever the different changes we will see in the different steps of the glycolysis, that will be the today's topic. Okay, so let's start. Now for these things, I'll take a hypothetical cells. Okay, just like this. Anyway, now so within the cells there are different types of the transporters. I have already said the blood transporters, by which, for example, is the blood transporters, by which we will see that the glucose that will be taken within the cells. So here we will see what the glucose that will be taken within the cells in the same way. Okay. Now whenever we are getting already the glucose over here, so there is a chances for the glucose molecule to go back again from within the cells to outside of the cells. That's in the first step. What we see, basically we know the glucose, what is the 6 molecules of the carbon, 1, 2, 3, 4, 5, 6. Okay. Now see the first steps of the reactions of the steps of in terms of the glycolysis, whatever the changes we will see, we just add one charged molecule along with the glucose. Whenever it will be charged molecule, that will be not easier for the whole complex of the compound to cross the membrane, because the charged particles, whenever we get in terms of the polar compound, that will be not able to cross the membrane. So that's why we'll write it over here. We'll get to what? For example, we are adding on phosphate molecules, so we'll get it over glucose 6 phosphate 1, 2, 3, 4, 5, 6, and for example, we are adding over here on phosphate molecules. Okay. Now see in the first step, from there, that's just to convert the glucose into glucose 6 phosphate, we can use the two types of the enzymes, or we can use the hexokinase. Another we can use the glucokinase. Now okay. Now the hexokinase is the enzyme that is present in almost in all cells. Okay, that is present almost in all cells. And what about glucokinase? Glucokinase that will be present in some specific type of the cells that I am going to talk. Okay. Now see, for example, if I just say that in terms of the hexokinase, let me write it over here first. That see the hexokinase that can react whenever the small amount of the glucose molecule. So whenever the glucose small in amount. Okay, but what about the glucokinase? Whenever the glucose molecules more and more in number, at the time the glucokinase enzyme that will be activated. If I want to show you in this way, for example, in terms of a graph, now try to understand from the graph. Okay, now see, for example, I'm just taking the graph over here. Okay, now see, for example, is the anaerobic performance. Okay, I'm using in this row, I'm just taking the performance of the enzyme. Performance of enzyme, and in this way I'm just taking the concentration of glucose. Glucose concentration. Okay, now see, whenever the glucose is coming during the feeding state, okay, for example, whenever the little molecules of the glucose, for example, in the slight change in the glucose concentration, whatever the enzyme that will be activated, at the hexokinase. So hexokinase that will be activated at it, there will see the rapid, just like this. Okay, in this case, try to understand from this graph. In this case, is what we will see the hexokinase, this one. You will see the rapid reaction. Okay, we will see the rapid reactions. Okay, and whenever it will go at certain level, just like this, it will be saturated. Okay, so rapid reactions, after that at a certain time we will see it will get saturated. So we'll see the saturation. After that, if we just increase the concentrations of the glucose as much we can, there will be no change in the what velocity, or you can say in the graph. So it will be just like this. If we just talk about in terms of the, if we just talk about in terms of the glucokinase over here, the glucokinase, it needs just like this. In terms of the glucokinase, this is in terms of the glucokinase. So if we talk about in terms of the glucokinase, here we need to know one thing, that the glucokinase needs more amount of the glucose for its action, more glucose for its action. Okay. Now there are certain types of the thing that should be clear in these cases. The first things we'll talk about over here, the Km and the Vmax. Km means, we have to know in this way, that the concentrations of the substrate, at which half of the enzymes will be saturated, the concentration of the substrate at which half of the enzyme that will be saturated. So in this cases, see, if I just say in these cases that in terms of the first, let's talk about hexokinase. See, it is getting saturated over here, just like this. Let me show the red one. It is the graph for the what? It is the graph for the hexokinase. So for example, half of the, let me show half of the enzyme. So half of the enzyme is getting saturated over here. For example, half of the enzyme getting saturated over here in these regions. Okay. So, and what about the glucokinase? If we say the glucokinase, half of the enzyme, for example, will be saturated over here, just like this. So this point, this and this point will be Km for what? This point will be Km for glucokinase, and this point will be Km for hexokinase. Okay, this will be the Km for what? Hexokinase. This will be the Km for glucokinase. Now see, what about the Vmax? Vmax means the maximum velocity. So in these cases we can write the K means the concentration of substrate at which half of the enzyme will get saturated. Okay. And Vmax means the maximum velocity. That means the speed of the reactions in this case. Now see, we'll get the Vmax up to here. We'll get the Vmax for what? This will be the Vmax for hexokinase. And what about the Vmax? For example, I'm just taking this part. This is the Vmax for what? Glucokinase. Okay. Now see, if I just compare in between the glucokinase and hexokinase, let's write it over here. If I write it over in terms of the hexokinase, so see the hexokinase in this both cases, if I just compare from the graph, the hexokinase having less Km and less Vmax. So it will have the less Km and less Vmax. Okay, and in terms of, if we just compare with the glucokinase, so let's write it over here, the glucokinase. Glucokinase needs more amount of the glucose molecules to get activated, to get saturated. Okay, so in these cases we can say the Km, that will be more. In terms of the Vmax, that will be also more. Now see, whenever the less amount of the enzyme is, if you just see the slight changes in the concentration of the glucose, what the hexokinase, that will be activated. That means the affinity for the glucose, that will be maximum in case of, or more in case of hexokinase. That means the affinity for glucose, that will be more in case of hexokinase. The affinity for the glucose, that will be less in terms of the glucokinase. Okay, so you can say from here that in these cases we'll see the affinity for glucose, that will be more. That means whenever the less amount or the slight changes in the glucose concentrations, we'll see over here the hexokinase enzyme, that will be activated. And what about the glucokinase? Glucokinase will be activated at the time when we will see there is enough concentrations, or we'll see the more concentration of the glucose over here, or we can say the number of glucose molecules is more over here. So in these cases we will see the what? In these cases we will see that the affinity for the glucose, that will be less. Affinity for glucose, that will be less in these cases. Okay. Now let's talk about, I have already said the hexokinase. Where we will see the hexokinase? Hexokinase, it will be found in almost all cells. See, hexokinase you can find in all cells. And what about the glucokinase? Glucokinase you can see in terms of the liver, within the hepatocyte, within the hepatocyte. And in some books it's mentioned that we can also get in terms of the pancreatic beta cells. Okay. Now see the hepatocytes. That means here we get the glucokinase. Now see, if we just see the structures of the liver over here, for example. Okay. Now within the liver, for example, is the hepatocyte, and within the hepatocyte, where it will be, that means the glucokinase will be where within the hepatocyte, the liver. If I just say over here, this is the nucleus of the what? This is the nucleus of the hepatocyte. Now within these portions, there is on top of the regulator protein, glucokinase regulator protein, that means glucokinase regulator protein. Within these regions, we will see there is a presence of what? Hexokinase. For example, this is the hexokinase. So we will see, sorry, the glucokinase. So where we will see the glucokinase we will see within the hepatocyte. Within the hepatocyte, where within the nucleus of the hepatocyte, we will get a protein that is known as glucokinase receptor, regulator protein, that is known as glucokinase regulator protein. Within what we will see there is the presence of glucokinase enzyme. Okay. Now see, whenever during the feeding stage, whenever the concentration of the glucose, that will be greater or will be much more. Whenever the concentrations of the glucose that will be more, the glucose that will come by the GLUT receptors or the GLUT transporter over here. By the GLUT transporter, we will see there will be the abundance of the glucose within the hepatocyte cell, and that will go over here, and that will activate the glucokinase enzyme. After that, we will see there will be what? Exertions of the glucokinase. The glucokinase will be activated, and it will just do what? It will convert the glucose into glucose 6-phosphate. It will convert the glucose into that means the glucokinase. Let me write it over here. Glucose 6-phosphate, just like this. Okay. So this was basically the comparison between the hexokinase and glucokinase enzyme. The first step. There are more steps. Okay. After that, we will get what from the glucose to glucose 6-phosphate. From there, we will get fructose 6-phosphate. From there, we will get fructose 1,6-bisphosphate. There are different types of the enzymes. One of the most important enzyme, that is phosphofructokinase. That is the vital enzyme in this process. Okay. And in this case, see, there is the unidirectional pathway. This is the unidirectional pathway, not reverse. It's the unidirectional pathway. So about the rest of the enzymes, I will talk about in the subsequent part of the videos. This is the glycolysis part 1, part 2. I have already uploaded a video on glycolysis part 1. Thanks for watching.